We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
Thank you for the invitation, and thank you to the amazing CARTA staff that helped put this together. So today I'm going to talk about a tool to, another tool, an additional tool to study human comparative studies with non-human primates. So there are a number of systems and models to study human evolution. You can look at the DNA. You can get genetic information by understanding and by sequencing the genome. You can look at postmortem brain tissues, so you can get insights into tissue organization and brain anatomy. You can look at the fossil records, and you can look at archaeological evidence. And you will get relevant information from all these systems. And what we over the years, what we wanted to contribute to those systems by bringing another tool to study evolution. And one way we plan to do that is to use stem cells. So we think that this is a missing link to understand studies of evolution. And we also think that you could use that to do some hypothesis validation, because now you have a live cell system that you can test, ask questions, and look at, for instance, neuronal behavior. So how is the brains of humans distinct from other primates? So just some of the differences are related to number of dendritic branches. So humans have increased dendritic branching. Humans have increased spine density, so the protrusions that come out of the neurons from humans there are more protrusions coming out, so there's more density in the spines. Humans have an enlarged cortex and also a delayed maturation, so that's also called neoteny. And that has been shown in that delayed maturation in many levels. For instance, humans have slow cortical growth compared to non-human primates. The cell cycle length is also prolonged, and there is slow dendritic spine maturation. So it has been proposed that this protracted development, also named neoteny, can be one of the mechanisms that have contributed to human-specific traits. So another way to look at neoteny is comparing humans to non-human primates. And here I'm giving you an example of a chimpanzee development. So that's I actually modified that from a graduate student that is a CARTA member, Shayla Steiner. So when humans and chimps are born, so the pregnancy time is roughly around the same. So it's about 8 months for chimps, a little bit quicker, and humans about 9 months. But when a chimp is born, it's already able to cling to mom. Humans are not. About 3 months old, humans can hold their head up, smile, hold things, maybe roll over, but chimpanzees can crawl, grab objects, and sometimes even start taking their first steps. At about 1 year, chimps can climb, can walk, can collect food, and begin their first social relationships. Humans may be able to, babies may be able to walk, may be able to say a few words. At about 3 years old, chimpanzees are nest building and grooming, and humans are starting to run, climb, but now they're also starting to count, form sentences. And then by 15 years old, chimpanzees are already having their first babies, but humans now have this even bigger myriad of things that they can accomplish and can do, including reading, writing, math, have a vast vocabulary, and can also have theory of mind by then. So humans develop much slower, much more slowly than non-human primates behaviorally too. So we wanted to use that the technology, that tool, to ask if there were differences that we could detect at the cellular level that would be relevant to understand brain size in neuronal maturation in humans, with the idea that the differences that we would observe can be captured at the cell level. So we're using a model system here. The implications of finding differences, of detecting changes, is that we could be able to understand some differences in susceptibility to infections, to neurological diseases, and even to cancer and even other things. And also that could help increase our basic understanding of the evolution of our own species. So we started at about 10 years ago by first developing those induced pluripotent stem cells. So those stem cells that we can develop from skin. So we change the program of a skin into a stem cell. And now that stem cell has pluripotency, so it can many potencies, and it can be differentiated into supposedly any cell in the body, including brain cells, neurons, astrocytes, which are the cells that we were interested in probing. So we developed those cells from humans and non-human primates. And what we did, our first question with regards to could we even observe that neotenic or differential developmental timing in vivo, in vitro, in the culture system, we differentiated those cells and we looked at how they changed over time in vitro system. And what we did that in a collaboration with Caterina Semendeferi's lab, and it was a work, quantification work done by Branca in her lab. We were able to detect that by measuring the sizes of the branches and the hybridization of the neurons over time, we were able to see that when you compare chimpanzees' neurons with human neurons, they were differentiating, or they were becoming more mature. They were making their branches at different rates. And what we were able to observe, and this is represented in this graph here on the top right, we see that chimpanzee neurons start the process of differentiating and branching like trees earlier on than humans. But then later on, humans pick up and differentiate at make more arborized neurons. So that is reproducing some of these aspects of delayed maturation in the beginning, but then reaching higher levels of maturation later. So we're able to observe that in neuronal behavior in vitro. Then our question was that in terms of function, so neurons are cells that are very specialized. They fire action potentials, which are electrical firing that they use to communicate to each other. So we have ways in the lab in which we can measure that activity, that neuronal activity, the neuronal conversation, how one neuron talks to the other, by using systems that are detecting neuronal activity by extracellular voltage changes. So those are electrodes that we embed into the culture, and the neurons that grow on top of it, when they fire action potentials, when they have this electrical signaling conversation, we can detect. And so we are able to not only detect the neuronal maturation by looking at the morphology, so the number and the size of the branches, but we can also look at neuronal function, how active the culture is. And in general, levels of activity, increased levels of activity are correlated with increased maturation rates. So what we see here is those CHs are channels. So that's a plate. Those little dots are electrodes. And those blinking sites that we see on the right top, they indicate if the neuron in the plate is firing, is detecting firing or not. So by doing that and looking at each channel where a neuron is sitting and it's firing, we can detect the rate in which each channel is detecting the neuron that is firing. So every trace here is indicating a firing event. So when we compare human neurons cultured on these different channels by listening to their activity to chimpanzees, and that's also true for bonobos, we can detect that in 2 weeks, chimpanzees and bonobos are firing more than humans. And that's quantified here on the side. But later on, at 6 weeks, we can see here that humans are firing much more than chimpanzees and bonobos. So we are detecting, bottom line, we are detecting this delayed maturation in both humans as well by morphology, by branching, as well as by neuronal activity. So our next question was, are there any genetic drivers for this phenomena that we are now able to reproduce in vitro? And what we, the way in which we wanted to check that is by looking at changes in neuronal regulation in genes, in gene regulation in neurons over differentiation time. So basically what we wanted to do is we wanted to look at neurons from non-human primates, and that we used rhesus, gorilla, chimpanzees, bonobos compared to humans, and we wanted to see what were the genes, what were the differences in gene expression that were unique to humans. So again, we did the same differentiation process that we did before. So we made neurons, and then we looked at those different species in comparison, and we want to look at principal component analysis with the previous talk. Brenna talked about that too. So it's a way in which you reduce all the information of the one individual in one dot. So each dot represents one individual at one condition. So what this is showing us is that in principal components place, we can reproduce phylogeny in that just by looking at gene expression, which is really important for us because it means that what we have in culture to some extent reproduces what we have in vivo because humans are clustering with humans, so that's the green dots. Chimpanzees and bonobos are clustering together, and chimpanzees are clustering together, and rhesus monkeys are clustering together too. So in general terms, our principal component analysis is separating species by phylogeny. Then we still wanted to ask what was unique to humans. So we performed a weighted correlation network analysis, which is a type of analysis that identifies the top upregulated genes in human-specific networks. And one of our top genes that we detected that was specifically differentially regulated in human during neuronal differentiation is called GATA3. And this is just an example of how the GATA3 expression is happening over time it's starting at lower, so that's a neuroprogenitors, those are the baby neurons. And when the neurons start differentiating, GATA3 goes up and stays up, but only in the green, which are neurons, the other ones, the other species, that gene is not regulating. So GATA3 is correlated with genes that are uniquely upregulated in humans. GATA3 gene is a zinc finger and is a pioneer transcription factor. So it's one of those genes that can bind to the DNA and to many locations in the DNA and change a lot of what the cell is doing. So those are usually genes that can do a lot of change and can change the fate of cells. So we thought this would be an interesting gene to look a little bit further. And we confirmed that the gene was expressed in humans and was not present in the other non-human primates. And we also looked at the expression in using the Allen Brain Atlas samples, which has brain samples from humans and rhesus monkeys at that time. And what we also noticed is that there is an increase here in red of the expression of GATA3 in human brains compared to rhesus macaque brains at the intermediate zone, which is developing area in the brain. So we confirmed that both in vitro and in vivo, this was also true, that GATA3 is upregulated in humans. We then wanted to know, since GATA3 is a transcription factor, it binds to the DNA, we wanted to know if it was binding to specific regions that were, for any reason in humans, were evolving more, were more differentially mutated in humans. So this would indicate that those areas could be, the GATA3 might be binding to areas that are accelerating in humans, in human genome. And to do that, we performed a technique called chromatin immunoprecipitation, followed by sequencing. So what we do, what this technique does is we're able to treat the cells with GATA3. So GATA3 would bind to specific areas in the genome, and then we can sequence the areas where GATA3 is binding. And what we can get from there is what DNA areas this transcription factor is binding, and how they look like if you compare them to the genome of chimpanzees, for instance. So when we do that, we confirm that GATA3 binding sites in human neurons are under positive selection. And what that means is GATA3 is binding to areas in the human genome that are not present in chimpanzee genome. So that tells us that this can be an important gene in human development, for instance. So GATA3 will bind to unique sites in the humans that are under positive selection. Then our question was, okay, if GATA3 is really important, what happens if I decrease expression of GATA3, what we call knockdown? So if we remove or if we decrease GATA3 presence during human neuronal differentiation, what happens? So that's a traditional things that molecular biologists would do. They would do gain or loss of function. And what we do that, you see here in black. When you remove GATA3, the transcription profile in principal component space changes. So, and we're comparing here black triangles with green triangles. What that means is when we collapse that graph, you can see here that both green and gray bars are GATA3, what happens when GATA3 is present. When GATA3 is absent, what happens is black, which means there is a change in the profile of what now are the cells are expressing to levels that are now look similar to chimpanzees. So the transcriptional profile changes of a cell that don't have GATA3 to being similar to a chimpanzee cell. And this is just an example of a gene that is changing. And that's a gene related to neuronal differentiation. We can, there are other genes too. So GATA3 regulates a component of transcription that distinguishes human neurons from chimpanzees. And the other thing that we noticed, and I'm just going to show this, my last data slide, when you do this, we remove GATA3, you increase the activity of the neurons to earlier times that they were before. So that means that now neurons are behaving, human neurons are behaving neotenically. So they are getting active earlier than they would if they had GATA3. Now they are closer to chimpanzee neurons. So GATA3 acts to delay the maturity of action potential. So when you remove GATA3, neurons become similar to chimpanzee neurons. So I talked about my conclusions in GATA3. In summary, it acts to delay the maturity of spontaneous action potentials, which is a key aspect of human neoteny. And we think that we can use this technology, human-induced pluripotent stem cells, as a tool to study human unique characteristics that can have a potential evolutionary impact. And with that, I just want to thank you all for coming, and also my collaborators, Rusty, Krishnan, and Katerina, and everybody else that work. It takes a village. Thank you so much.